Hello everyone, welcome back. This is Dr. K. Geeta from Department of Biotechnology, Kamaraj College of Engineering and Technology, Virudunagar. Let us discuss a little bit about antibodies, their structure and their function. So, this is what an antibody looks like, as you can see. Discussing about its structure, antibodies basically are made up of two light chains, as you can see here, and two heavy chains. In the light chain and the heavy chain, the difference is in their weight. The two light chains, they are identical to each other, approximately 25 kilodaltons in size, and the two heavy chains that we have are also identical, but the size is approximately 50 kilodaltons in size. And if you look at the structure, both the heavy chain and the light chain, they comprise of a variable region and a constant region referred to as VL, VH and CH and CL. And in this, the first 100 amino acids usually forms the variable region of both heavy chain and the light chain. And each of the light chain is bound with the heavy chain with the help of disulfide bridges, and the heavy chain is also bound with each other through disulfide bridges. And there is a special region in the beginning called the complementarity determining region. This is the area where the antigen comes and binds, and other than this, the remaining structure is almost similar in most of the antibodies. And before we delve into the structure in detail, a very important aspect with respect to antibody is digestion of the antibodies with different enzymes. So, if you see with papain and with pepsin and in presence of mercaptoethanol, the results are quite different. When papain is used for digestion of an antibody, you will see the three fragments that are obtained. Out of the three, two are just the Fab and the one is the FC fragment. Fab a fragment is obtained with, this is the antigen binding region, and this is found to be crystallized in cold storage. Whereas at the same time, when pepsin is used for digestion, the entire FC region is completely degraded, leaving behind the two Fab regions. And irrespective to these two, when mercaptoethanol reduction is carried out of an antibody, you will find that it will eliminate all the disulfide bridges, leaving behind only the heavy chain and the light chain as individual compounds. So, this is the result of papain, pepsin and mercaptoethanol reduction. Okay, coming to the structural details of an antibody. If you look at the structure, it has repeating domains of around 110 amino acids, and as I have told you, there are heavy chains and light chains. In the heavy chain also there is a variable region, or just one variable region in the beginning of around 110 amino acid, and then you have 3 to 4 constant regions depending on the type of antibody we are discussing. And as for the light chain, there is always one variable region and one constant region. And other than this, there is also a hinge region, which is the one basically which allows the antibody structure to move and interact with different antigens. So, this what you are seeing is the hinge region, and this region has been found to be rich in proline residue, which gives the flexibility to the antibody structure. And hinge is not found, this region is not found in all antibodies. You will see that IgG, IgA and IgD are the only antibodies where you will find the hinge region. And fluorine residue is the residue which are targeted for proteolytic digestion when papain and pepsin is used. And other than this, it is also found to be rich in cysteine residues, which forms the disulfide bonds. And as I have informed, IgM and IgG do not have this hinge region. Instead, they have something called an extra heavy constant region, as I have told you while discussing the heavy chain. So, this is the overall picture of antibody structure. Coming to the classes of antibody, there are 5 basic sequence patterns that are obtained with respect to the antibodies. They are alpha, gamma, delta, epsilon and mu, and based on these patterns, antibodies are classified into 5 classes which are known as IgA, IgG, IgD, IgE and IgM, and these are called as the isotypes. Please remember, the isotypes are the 5 classes of antibodies based on the heavy chain. And each of these class can have either of the 2 types of light chain, that is the kappa light chain or the lambda light chain. So, this is about the classes. Now, based on further minor differences, IgA and IgG have been further classified into IgA1, IgA2 and IgG1, 2, 3 and 4. These are the subclasses based on further variations. Other than this, you will also find B cell receptor which also have the similar structure as you find in case of B cells which are found on the surface of on the surface as B cell receptors, which are actually the MIG. And these have the difference between your regular immunoglobulin and the B cell receptors are they have a very short cytoplasmic tail and they cannot transduce signal as is carried out by your antibodies. So, we saw that there are 5 classes, right? So, what are the differences? How are they different from each other? So, if you overall look at the structure of any antibody, the differences lies in the number of subunits present, the number of domains present, the number and location of various carbohydrate chains that are available, then presence or absence of the hinge region, and also based on the number and location of different disulfide bridges which are available in an antibody structure. So, structurally this is how different antibodies look like. So, talking about each one in little more detail, you can see here this is the structure of an IgG and we have spoken about the hinge region. This is the variable region. This is where your CDR or the domain where the antigen comes and interacts exists, and these are the constant regions, and this is the Fab and this is the FC region, and this is the most abundant antibodies which is found. Approximately 80% of your serum immunoglobulin is comprised of IgG, and the concentration being very high, approximately 10 mg per ml of the blood of the serum, and there are 4 subdomains, subtypes, out of which each one has different functions and each one has different slight variation in their structure also, and these are known to be secreted as monomers. Then coming to IgM, IgM is the most unique structurally among the 5. These exist as pentamers and they are also found in the serum, but at a very low concentration, around 5 to 10%, and usually these are the structures which are also expressed on the surface of the B cells called MIgM. And this is the first immunoglobulin which comes into picture as soon as there is an antigen available in the body. So, these are the primary immune response immunoglobulins. Then coming to IgA, and this particular antibody is quite unique in its structure. If you see, they exist as dimer, and the 2 molecules, they are joined by something called J chain, which is unique when compared to other 3 types. J chain is also found in case of IgM, which is known as the joining chain, which is the one which holds the different monomers in place together to form the actual antibody. And these are found at a slightly higher concentration than IgM, around 10 to 15% of the serum IgG. Serum molecule is serum immunoglobulin is IgA, and 2 IgA molecules, they are always joined together through the J chain. Then coming to IgE, which is the lowest in secretion, and whenever there is an infection or whenever there is any kind of particulate matter coming into the body, these are the ones which are released in response to the immune reaction. So, under hypersensitivity reaction. for example, asthma. So, IgG level, if there is an increase, you can correlate it with some kind of an immune reaction that is happening in the body. And these are the ones which binds to mast cell and basophils through Fc receptors. And the final one is IgD, which are usually expressed on the B cell surface, and both IgM and IgG, they are expressed on the surface of the B cells, and these are the ones which are usually involved in antigen processing and presentation also. And these have a very low serum concentration, you see, when compared to other molecules like the other antibodies, the concentration being 30 micrograms per ml. So, to conclude, you can see here the different types of antibodies. Structurally, IgA and IgM are the ones which exist as dimer and pentamer, whereas IgE and IgG, they are secreted as monomers. IgD secreted form does not exist, and these are basically the function is naive B cell antigen receptors. So, overall, to conclude, we have 5 different classes of antibodies, each one differing in their structure. Superficially, if you look at all the antibodies, they have heavy chain and light chain with variable and constant region. So, with this we conclude the topic on antibody structure and function. Thank you.